Climate change is a phenomenon that has come to technically disrupt our food system and impact mostly on our ability to be able to produce more food. As you know, Zambia's population is increasing, so there are two things here we're addressing: a population that is growing and our ability to be able to grow food to sustain that population. And climate change is one of the challenges, that we are facing as we strive towards producing more. How are we addressing, the issue of climate change as ZARI? Firstly, ZARI as a department, a technical department, I must mention, in the ministry, is charged with the responsibility of developing soil and crop technologies with a view of contributing to increased food security, also job creation and overall economic growth. Now, in the area of crop improvement, we have been developing various crop varieties that are suitably adapted to the ever-changing climate. So, for example, in the last 5 years alone, we've developed over 105 climate-smart varieties. These range from maize, sorghum, cowpea, cassava, and several other crops, that are, either suitably adapted to an ever-changing, climatic condition. So some of the traits that we focus on in our breeding efforts include selecting varieties for drought tolerance. As you know, one of the impacts of climate change is reduced rainfall or intermittent dry spells, or even extended or prolonged dry spells, which you can collectively maybe characterize as a drought. So what our scientists have been doing at ZARI in our various ZARI stations dotted across the country is they've been selecting, crop varieties that either mature very early or varieties that, again, like I said, are able to utilize less water but be able to yield or produce enough food or enough grain or enough edible parts for our population to consume.
So the unit basically looks at focusing on microbes that are existing in nature and bringing them to the lab, and we're able to multiply them and reintroduce them to the environment so that they can be used for the improvement of soil health, soil fertility, and just improving crop productivity. So what we've focused on mainly is evaluating various strains, various bacteria, and looking for the suitable ones for each crop that exists in the various regions of Zambia. And we know that we've got 3 agroecological regions. We try to look for species of bacteria that are crop specific. For instance, right now we're focusing on soybeans, maize, common beans. These 3 crops have been used in these evaluations.
Research activities that border on soil and water conservation, and also research that tries to manipulate the soil by means of drainage, tillage, or aeration to improve crop productivity.
When we talk about plant tissue culture, this is like growing plants in a controlled environment. So you get them from the outside and grow them in the laboratory where you're able to control the environment. And the advantage of tissue culture is that you'll be able to propagate more plants in a small space. So that's one of the advantages as compared to the outside environment. So the plants that we obtain from the outside environment is brought into the lab, and then we use the parts of the plants, that could be the shoot tips or the nodes, in order to multiply the plants. So before they are brought into the laboratory, we actually index them to make sure that there is no any virus diseases, so that what we are multiplying is clean plants.
The oilseed breeding program, we are dealing with a number of crops, and these include sunflower, sesame, soybean, and safflower, as well as castor. Yes, we are breeding in line with the theme for this year, adapting to climate change. So what we are doing is that we are breeding materials that are tolerant to drought. First of all, drought is one of the key issues that we are dealing with, because the active breeding programs that we have currently is sunflower and soybean. So for both crops, we're looking at materials that are tolerant to drought, and then materials that are also tolerant to diseases and pests, because as you are aware, the climate change has come up with a number of new diseases as well as new pests. If you talk about soybean, you know there's the issue of soybean rust right now. So we are trying also to screen our materials for tolerance against soybean rust. Then also we also want to help the small scale farmers. We are breeding for materials also that are high yielding, and then materials that are tolerant to pod shattering, or materials that delay to shatter for the sake of our small scale farmers.
And in line with this year's theme, which is adapting to climate change, our team fits in very well, because you know that trees play a very important role in terms of climate change mitigation. Trees act as carbon sinks that absorb the carbon dioxide in the atmosphere, which is responsible for the depletion of the ozone layer, and hence the problems that we are facing as a result of climate change shocks. So in line with that, we are encouraging our farmers. First, when they come here, they'll be able to get the different seedlings or fruit trees. Later on, apart from just raising them as orchards, we are encouraging them to integrate these fruit trees with other annual crops. We understand that for our farmers, sometimes they're a little bit impatient to wait for a tree to get up to so many years before it starts fruiting. But when you come here, you find researchers that have developed different propagation techniques that reduces the maturity period of these fruit trees. Within 1 year, 6 months to 3 years, they start fruiting. But before they get to that fruiting part, you can still use that orchard to integrate with other annual crops, such as the legumes, beans, cowpea, soybeans, and the likes. And all this that we are doing is fitting into our, ministry's policy of crop diversification.
The gene bank conserves various crop diversity that are tolerant as well as resilient to climate change. So most of these genetic resources is used as an adaptation strategy as well as a mitigation to climate change. And most of our program is based off the fact that we of course collect throughout the country, and of course we promote the utilization through on-farm conservation, as I had earlier indicated. And this on-farm conservation, we take these materials to the farmers where they conserve these materials in their fields. Hence the genetic resource in that regard is adapting to the prevailing climate change.
To generate and adapt post-harvest appropriate post-harvest technologies in order to facilitate reduction of post-harvest crop losses caused by storage waste, as well as enhancing the broad utilization of crop products. So those are the mandate of the Food Conservation and Storage Unit. So we have gone further a little bit to promote and upscale such technologies which are known as hematic storage technologies. So we have these hematic bags. Now these are bags that don't use chemicals. They are based on the principle of suffocating the insects. So they have 2 plastic bags inside them, and then when you put the grains, you just close them properly, and you can have your grain protected. The quality is maintained, and the grain is not attacked by the pests. If there are any pests that are in the grain, they'll be suffocated. If the eggs are there, the new insects will get suffocated, and then they die off. And the advantage is that these bags, you can use them over 2 or 3 seasons.